Dear students, today we will study an important genus from fungi, that is Puccinia. And like that of the previous classes, here also we will discuss about vegetative structure and then elaborately reproductory mechanisms of Puccinia. On exam point of view also, this Puccinia is very important because it may ask for essay question, 4 marks question. You can expect even 1 and 2 marks questions. Many of them is normally asking from this particular chapter. So, first we will discuss about its systematic position, and it is coming under the class Basidiomycetes, subclass Heterobasidiomycetes, order Uridineales, family Pucciniaceae, and of course genus Puccinia. And I will repeat once again, that is class Basidiomycetes, subclass Heterobasidiomycetes, order Uridineales, family Pucciniaceae, and the genus Puccinia. First we will discuss about its habit. And the genus Puccinia, here we are discussing about the species Puccinia graminis. Okay, and here on spelling mistake is there, that is not a spelling mistake, G is capital, and remember always species name starts with a small letter. And this genus comprises about the Puccinia genus comprises about nearly 3000 species, of which 147 have been reported from India. All are internal parasites. That means that is normally the hyphae of this genus is normally found inside the host tissue. So, it is internal parasites, especially on angiosperms. Only the spore masses, so when it is doing reproduction, it will produce spores, and these spore masses are visible externally. So, the different species of Puccinia cause different types of rust diseases in wheat itself. And among them, for example, black rust is due to Puccinia graminis triticae, and this particular species we are going to study in detail today. And we can see yellow rust is due to Puccinia striformis, and it is a new name is for the species striformis, that is Puccinia glomerum. And the brown rust or leaf rust is due to Puccinia tritricina or Puccinia recordita. So, anyway, today we are going to study in detail about the Puccinia graminis. So, first we will discuss about its habit, and the Puccinia graminis is an obligate parasite. What is the meaning of obligate parasite? Already that definition for obligate parasites is written here, that is, it is a specific host pathogen relationship is there. That is, it can only grow on a particular host. Then it is called a obligate parasite. So, Puccinia graminis triticae is the variety name. Triticae is an obligate parasite, and it is polymorphic. That is, what is the meaning of this polymorphic? Because to complete its life cycle, it produces many spore formations. That is, Puccinia graminis triticae is a polymorphic organism because it produces 5 different spore forms in its whole life or life cycle. They include iridospores, teletosaurs, it is otherwise called a teleospores, Telio, teleospores, and Basidiospores, it is otherwise called as Sporidia, 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 and Pycnospores, it is otherwise called as Spermatia, and Basidiospores. Okay, and also this particular species is considered as, its life cycle is considered as macrocyclic, because in order to complete its life cycle it is passing on different types of life forms. So, it is macrocyclic. And because the Uridia or iridospores, sometimes you can say as Uridinia, and Telia or Teletosaurs are produced on wheat, which is the primary host. So, the Pycnidia or Pycniospores or Spermatia and Assia or Acidiospores are formed in barberry plant, which is the secondary host or alternate host. When Teletosaurs germinates, Basidiospores formed in the Promycelium, that is, it is not with any spores. It is a very complicated species. So, try to study in detail about this, how these 5 types of spore forms are producing in this macro life cycle of Puccinia graminis triticale. Okay. So, in this particular slide, it is already written here, that is, it in the wide range of hosts including wheat, barley, oats and rye. Okay, that is in case of this Puccinia graminis. That particular variety, that is Puccinia graminis triticale, is only involves or infecting wheat plant. Okay. So, the Puccinia graminis triticale involves in its life cycle 2 distinct alternate host plants, that is wheat, that is Triticum vulgare, and the family is Poaceae, and barberry, that is Barberries vulgaris, family Barbaridaceae. Okay. So, that is related to the introduction of habit description about this Puccinia graminis. And here we can see this wheat and the barberry. That is black rust disease is due to the infection of this particular Puccinia graminis. Okay, some of the species of this barberry produces edible fruits. And what are the symptoms? This is all part of the general part. The symptoms of the disease occurring in wheat includes large elongated and brown pustules, that is Iridosauri. And how this Iridosauri is forming, producing, we will study in detail on the stem, leaf, sheath and leaf. Later on, these brown pustules change to black colored large pustules, that is due to the presence of Teletosauri. Sauri means sporangium plural, it is sauri. Grains of the infected plants are shriveled. In Malayalam it is padira, much lighter in weight and thus reducing the yield. So, this due to the infection of the Puccinia, it won't kill the plant. But as an economical yield related to the wheat production, the quality of the wheat reduces. That is a problem. Okay. Otherwise, the pathogen shows a balanced host parasite relationship. Even in the severe infection, the parasite does not cause much serious damage, except that growth of the plants may be somewhat retarded and the grains may be reduced in size and poor quality, and it won't kill the plant. Okay. And a country like India, our staple food is cereals, that is wheat and rice. So, normally it will create so many such related, that is the problems related to the famine that may be caused due to the presence of this infection of this Puccinia graminis. Otherwise, as part of the plant host relationship, it won't show any serious symptoms. Okay. And on barberry, infection first starts on the dorsal surface of the leaf, that is the lower surface. See the lower portions of the leaf in the form of minute dark colored flask shaped pycnia, and which appear as yellow spots. Beneath pycnia on the ventral surface, that is this figure, ventral surface, appear cup like projections of Assia or Basidia or Basidiospores. So, how it is occurring we will study later. And first we will study about the life cycle on wheat, how this infection is occurring in the first primary host, that is wheat. There we can see the presence of two types of spores, that is uridospores and teletosaurs, remember. And anyway, in the first paragraph, that is this uridosauri, that is the retrust stage or the first infection is as a result of the presence of a particular spore. That is, first spore infection is due to the presence of Basidiopores. How the Basidiopores are produced, we will study later. Now you just simply understand that the first infection is caused due to the presence of a particular host, particular spore infection, and that spore is Basidiopore. Okay, and it is brought from the infected barberry plants or by the uridospores themselves coming from the neighboring wheat plants. Okay, anyway, we will study in detail. So, first this is the cross section of TS of the wheat leaf, and this is the stomata, and guard cells are there, substomatal space is there, and there on the surface we can see this one spore is there, and that spore is Basidospore or Basidiospore. And after falling on this leaf surface, it will absorb moisture or water, it starts to germinate. And see their dicaryotized condition we can see in the spore. That is, two nuclei is there, and through the germ pore it starts to produce a hyphae. That first hyphae we can say as primary germ tube, and it will produce a bulbous portion which is called Presorium. And Presorium after the formation, here it is written that is the germ tube grows over the surface of the epidermis of the host, and on reaching the stoma is the singular of stomata, its tip swells up and forms a vesicle-like structure called Presorium. Here we can see this Presorium. Okay, right? And the protoplasm of the germ tube migrates into the Presorium. And after the formation of this vesicle or balloon shaped Presorium, we can see the protoplasm is migrated towards this Presorium. Now it is cut off from the germ tube by the septum. So after the Presorium formation, the nucleus along with the cytoplasm all comes to or rushes to the Presorium. Then it will form a wall near to this germ tube, primary germ tube, and now there is no need of this spore. All the internal protoplasm and the nuclei are now present in the Presorium. Okay. The protoplasm of the germ tube migrates into the Presorium. Now it is cut off from the germ tube by a septum. Then the Presorium produces a narrow hyphae. It enters into the substomatal chamber through the stomata. See, now it produces a substomatal vesicle. From that we can see the as a result of continuous mitotic activities it produces the hyphae. And see, there also we can see this dicaryotized condition because it is developed from a dicaryotized cell, that is that particular spore. See, and likewise we can see the first infection on wheat plant. Then how this, from this infected hyphae, how the first spore, that is the uridospores, are how it is produced. Within 5 to 6 days, the mycelium absorbs, that is the dicaryotized mycelium, absorbs sufficient food from the host. It begins to aggregate near the surface of the infected organs and forms a compact mass. That is why it is written like this, and many of you are not with the reference books, so you can use this particular points as notes. Okay, and what happens here after 5 to 6 days, within that short period itself, we can see all the intercellular spaces of the leaf. Because of monocots, there is no palisade tissue, only the spongy tissue is there, and in between the intercellular spaces we can see the complete mycelium mass, dicaryotized mass of tissue or hyphae. Now it is there, and it form a compact mass, and these then it produces near to the surface of the infected leaf, near to the epidermis, it produces spores which are called as uridia. Okay. And this uridia arise vertically in the layer of binucleated parallel cells known as basal cells. So, all these hyphae are dicaryotized. Developmental stages are not necessary. Anyway, we can see near to the epidermis it forms some basal cells, and these basal cells are also with a two nucleated condition, and from that it produces vertical upright hyphae. And the upper cell, such produced upper hyphae or vertical hyphae, divides again, and its upper daughter cell swells to form a single binucleate oval iridospores, because all the hyphae are dicaryotized. So, the spore produced is also what? Dicaryotized, that is iridospores. Okay. So, thus the iridospores are formed in a group, and each such group is called a iridospores, or collectively we can use the bunch of iridospores as uridinium. Clear? So, the developing iridospores exert pressure on the upper epidermis, and as a result it bursts out, extending or protruding through the broken area, and now the iridospores get exposed. This stage is called a red rust stage. At this time, that is after infection, within 1 week, we can see the complete field is with this magenta or red colored small patches, and it is due to these many events that spores, iridospores or iridosauri, which is produced after 1 week. And see this is the actual figure, and here this is the structure of iridospore, and this is the line diagram, and it is the cell is and then two layered. The outer layer is with ornamented pricky structures, and what we can see binucleated condition, but here it is one, that is after the here in down figures we can see the two nucleated condition, but here and there we can see karyokinesis is occurring. Okay. And in this particular stage you can draw as binucleated condition or single nucleated. Single nucleated means it is here, it is, and the nucleus becomes diploid, that is fusion. Okay. And this is a structure of this hyphae, and the hyphae exceeds, that is the basal cells and upright upright hyphae which produces the spores which are called binucleated iridospores. The 2 marks question you can get, that is what is the structure of iridospores. Then we should write it is 2 layered protective cell wall is there, cytoplasm, and dicaryotized condition. After some period it fuses and form a diploid condition, and sometimes it is appearing as dicaryotized itself. And here it is the actual structure of this iridospores and this sore. Clear? And how these teletospores are produced, that is after 1 week we can get red colored patches, that means formation of the iridospores. Later in the growing season, after some period of such infections and production of iridia or iridinia or iridospores, the same sore itself, it starts to develop the second host, and it is the teletospores. And the teletospores are at first developed among the iridospores in the same sorus, and they are dark brown or black in color. Gradually, as the season progresses, more and more teletospores are produced, whereas the number of iridospores is reduced. Finally, the sorae contain only teletospores. That is the second stage in the primary host. That is the same iridospores. First, we can see the infection spreads. That is, iridospores are again infecting the same plant and forming again and again iridospores. And towards the end of the season, the same iridosauri turned into what? Teletosauri. Clear? And gradually what happens? Only we can see the same sorae, it is with the only teletospores. These sorae are called teletosauri. The teletospores in the teletosaurus exert pressure on the overlying epidermis, which is at first lifted but finally ruptured. The black teletospores are now exposed. This stage is called black rust stage. Red rust stage, now it is turned into, towards the end of the season, it has turned into black rust stage. And see, this is the diagram. This is the actual section of the leaf. There we can see the sorae, but the spores are with minute differences. See the structure. And this is the teletospore. It is two-celled. That is the main difference. It is two-celled, spindle shaped, and has a thick, smooth, dark wall. Okay. So in the teletospore, each cell is binucleate. Each cell. That is, some cases the iridospore is appearing as bicelled. Here also we can see this bicelled condition sometimes. Very rarely we can see the fusion, very rarely. But otherwise, here also we can see the each cell, that is the teletospore is spindle shaped, bicelled, and each cell is binucleate. The germ pore is terminal in upper cell and terminal in upper cell and it is terminal in that is, it is bicelled, and from both cells we can see the it may develop into hyphae. That is the terminal here in C. Under C we can see this upper cell is the terminal portion, and so the terminal portion is with a germ pore, and the lower portion, lower cell is also we can get. That is, upper cell is with a lateral, the germ pore is terminal in the upper cell and lateral in the lower cell. Okay. So the two nuclei fuse in each cell, forming a diploid nucleus. And here we can see this fusion stage. So lower cell is undergoing fusion, the upper cell is already fused. Okay. And becomes diploid. Here actually we can see the karyokinesis. Okay. So that is the this is actually the towards the end of the life in the primary host. That is a host infection. Host is infected by the first Basidiospores, then it formed the Presorium and the hyphae formation, and formation of the first sorae, that is iridosauri, and iridosauri, that stage is called red rust stage. Then slowly towards the end of the season, the red rust stage is turned into the black rust stage, that is the formation of the teletospore. See the difference between the iridospores and teletospores. Okay. And iridospore is single celled, here the teletospores is bicelled. Both the cells are with a binucleated condition, and towards the end of the development of teletospore, we can see the fusion process or karyokinesis, and as a result the teletospore becomes diploid. And here outer surface is smooth, but in iridospore it is pricky. Okay. So then what happens? The next stage, that is basidial stage, and here it form after a period of frost period, and under favorable condition the teletospores germinates in situ, that is within the wheat plant itself, in situ, to produce a basidial stage in the life cycle. And this characteristic structures of this stage are the Basidia and Basidiospore formation. Here we can see this that is this it undergo meiotic process and form this. Here we can see this pollen tube germ tube formation, that is the terminal cell is with the upper part and that is lower cell is with a lateral germ pore formation, it is, and it has diploid a cell already and it undergo meiosis and form 4 cells. Among the 4 cells, 2 we can consider it as plus strain and 2 minus strain. Okay. And like that of the any Basidiospore formation, here also we can see the sterigma or stigmata formation and the bulbous structure, and like that of the previous chapters how the Basidiospores are formed. Likewise here also we can see the formation of Basidiospores, and it forms sterigma and balloon shaped structure, and through the sterigma or the tube portion the cytoplasm and nucleus enters, and when it is matured it cut off the sterigmata and the germ tube and releases the Basidiospores. Likewise the Basidiospores are formed. Okay. And 2 out of the 4, here it is written again, that is what portions just I discussed, that is 2 out of the 4 Basidiospores on which epibasidiums are the plus strain and the other 2 minus strain. Epibasidium means nothing but it is the young stage of Basidia, and they are small unicellular uninucleate haploid structures. When mature, the Basidiospores are discharged by water droplet method or spore drop mechanism. That is, it is nothing but through normal raindrops it will release, and they are carried by wind to the leaves of alternate host barberry. Normally in wheat plantations nearby area in North India especially, we can see plenty of barberry bushes. So normally it can get a secondary host. So when the Basidiospores fall on these secondary alternate host, that is barberry, we can see the life cycle, the second stage of the life cycle we can see. So the Basidiospores remain viable only for few days. They cannot bring about infection to wheat and thus perish soon. If it is not falls on a alternate host, if the host is not available, it normally perishes. And then next we will study about the life cycle on barberry, and this phase is also haplophase, because already in formation of the Basidiospores it becomes again the spores becomes haploid. Then this phase in the life cycle is confined to the alternate host barberry, that is Barberries vulgaris. It starts with the Basidiospores and the haplophase consists of primary haplomycelium and 2 spore stages. Here the juvenile mycelium which is emerging from the Basidiospore is called a haplomycelia. Then we should study about 2 stage formations, 2 spore formations, that is pycnidial spore and acidial spore, okay, or spermagonial stage or SCL stage. Okay, already 3 stages we studied. What are they? Iridospores, teletospores, Basidiospores. 2 more spores are there. Then haplomycelium, already I told, that is the Basidiospore germinates on the leaf of the alternate host. If the moisture and temperature conditions are favorable or suitable, it puts out a germ tube called primary hypha. This infects the young leaf of alternate host normally piercing through the epidermis. The cuticle of folded leaves, it is, see this Barbary is the dicot. Older leaves is due to thick and allow penetration by the delicate germ tubes. So it normally infects that is younger leaves. It will infect younger leaves. Once within the host tissue, it grows vigorously and branches freely to form primary mycelium, also called monokaryotic or haplomycelium, because here it is only a single haploid nucleus. But in wheat plant it is, even though it is haploid, but it is dicaryotized. Here it is monokaryotic. Okay. Then first stage is called a Sparmagonial stage. About 4 days after infection, the hyphae of the haplomycelium begin to collect beneath the upper epidermis like that of the wheat. They form a dense mat. Small light patches on the upper surface of the Barbary leaf are the external signs of the infection at this stage. And here we can see this infected condition. The mycelial hyphae ramify or branched in the intercellular spaces between the mesophyll cells of the leaf. They produce hostoria, which penetrate the cells of the host tissue. Hostoria is nothing but it is the absorptive hyphae which absorbs nutrients. For the growth of the hyphae, it may often happen that several Basidiospores of different strains infect the same Barbary leaf. So remember, the Basidiospores are of plus strain and minus strain. Naturally, the newly formed haplomycelia will be of two different strains, plus and minus. Okay, because at the same time we can see the infection of both the strains. They develop side by side. The two strains of mycelia can thus coexist in the same leaf, plus hyphae and minus hyphae. Even if they are very close to each other, no fusion occurs between them in their early stages of development. Consequently, each mycelium remains haploid for some time. Fusion may occur later. And what are the normal conditions of this particular infection? So here in brown patches we can see the uninucleated hyphae, and it produces normally when it starts to develop spores. The primary mycelium, this first formed mycelium turned into different types of hyphae. Okay, before the formation of Pycniospores or Spermatium, we can see the hyphae is formed into different kinds of hyphal structures. Now we are going to study about that. So from this mycelium mat arise groups of hyphae which appear as small flask shaped structures called Sparmagonia, also called Pycnidia. So the Pycnidia or Sparmagonia, like the mycelium from it appears as the mycelium like that of the normal mycelium, but it is and are of plus or minus strain. Okay, and from where it is growing, then from plus Basidiospore it will be plus. If minus, then it will be minus. When Sparmagonia mature, the infected areas become swollen and seen as small orange yellow bumps on the upper surface of the infected leaf. Okay, and the Sparmagonia are subepidermal, that is just below to the epidermis in position, and they are buried in the mesophyll tissue of the area, which gets thickened as a result of the pressure of the fungal presence of this thick fungal mycelia. Each Sparmagonium consists of wall surrounding the cavity, that is pseudoparenchymatous hyphae, and it opens on the upper surface of the host leaf through a small pore called ostiole. Then what are the special hyphae present in this sore, that is in this Sparmagonial area? It is nothing but This Basidiospore of plus and minus strain develops to form fungal mats in the same leaf, and it produces clumped structures just below to the Barbary leaf epidermis and form yellow bumps, and that area is called as Sparmagonia. Okay. And first, what are the individual hyphal forms present in the Spermagonia? First one, spermatiophores. That is numerous fine elongated uninucleated cells or short hyphae arise from the cells of the wall. They project into the cavity of spermagonium and which are called as spermatiophores. Okay. So, they are closely packed and arranged in a palisade like layer. That means closely packed elongated cells. That is why it is palisade like layer lining the cavity. Each spermatiophore by successive divisions of its nucleus form or obstructs at its tip, like that of the conidio formation, number of small cells or spores, number of small cells they will produce, and these are called Spermatia. And each Spermatium has a single nucleus and very little cytoplasm. The mature Spermatia fall into a spermagonial cavity. They are non-motile and cannot infect either host. See, this is the condition. But now we can see this figure, and here we can see this this palisade like hyphae, closely packed uninucleated cells, and these are called spermatiophores. Why it is called a spermatiophore? Because it is generating the spermatia. Okay. So, these are the palisade tissue centrally located, and it opens through an opening, and it is called ostiole. Okay. And here and there we can see a sterile hyphae is also there, and it is called a paraphysis. In some reference books it is called a periphysis, and both, what is the meaning? It is the sterile hyphae which is produced among the spermatiophores. Okay, that is a second type of hyphae present in the this particular sorus or sporangia. Okay. And the third one is the receptive hyphae, or it is the flexus hyphae. Okay, that is the sidewise, it is actually protrudes through the ostiole, that is called a receptive hyphae. Okay. And first here it is spermatiophore. These are the explanation for spermatiophores. These are palisade like tissue which is closely packed, and it cut off normally the uninucleated spores or cells which are called spermatium. Okay. And normally the spermatium is not able to infect any Barbary leaf. Okay. So, the spermatium, this spermatia, it is actually otherwise called Basidiospores or Pseudospores. So, spermatium is produced, and here it is that how the spermatium or Pseudospores are developing. The 1, 2, 3, 4 that stages, that is first one, it is the spermatiophore, and slowly it do mitotic divisions and form the small spermatia, and it is continuously developing spermatia. Okay. And next, periphysis or paraphysis. They are long, delicate, sterile hyphae. Okay. And normally they carve outwards in a cluster towards the ostiole. Finally, they project through the beyond the ostiole portion. Okay. It is also, it is nursing in function and also act like a shock absorbers. So, nutritive and protection. Then the third hyphae is flexus hyphae or receptive hyphae. So, adjacent to the ostiole and below among the tapering among the paraphysis or periphysis develop another kind of hyphae. They are slender, delicate and cylindrical with blunt free tips. It won't produce any spores, but it is dilated. It is with the dilated tips. So, being flexus, these are named the receptive or flexus hyphae. They are septate and may be of simple or branched, and each septum has a central pore. That is, see the general class we studied about the spores, types of spores. That is, even though the hyphae is septate, it maintain a continuity due to the pores in the intersecting walls. Here this Flexocyphae is with such pores. That pores are very somewhat larger central pores. Okay, so the when the Spermagonia or Spermatia matured, and how this development and the Spermatia, about the nature of Spermatia is with a different opinions among different fungal scientists. That is, it was in 1927 that the mycologist named as Craigie, Buller and their associate demonstrated that the Spermatia function as male cells. They are considering the Spermagonia or Spermatium or Pycniospores are male cells. So the receptive or flexuous hyphae are supposed to function as trichogyne hyphae. Trichogyne hyphae means attaching or act like a passage hyphae, trichogyne. They develop late and are spermatized by the Spermatia of opposite strain. Spermatization means it is nothing but this is a plasmogamy, that is a fusion of cytoplasm. Okay, so that is about the receptive hyphae. See the figure once again. That is, here we can see this three kinds of hyphae which are present in this Spermagonium. That is Spermatiophores, Pycniospores or Spermatia, then receptive hyphae. Okay, receptive hyphae sometimes it may be branched also with the dilated ends. Okay, and then next stage we will study about how this spermatization occurs, and it is nothing but it is a mechanism of plasmogamy in Puccinia, and it is known as spermatization. The mature Spermatia exude from the ostiole of Spermagonium in a drop of sticky thick liquid called nectar. That is, when the spermatiums are formed inside the flask-shaped cavity, it produces a sugary honey-like structure or nectar, and it forms a glossy drop over this ostiole. Okay, then the nectar with its scent and sugary taste, which normally attracts insects. Normally flies attracted towards the honey and to the leaf, and the Spermatia stick to the legs and the proboscis or antennae of the visiting insects, and thus are dispersed from leaf to leaf or from one Spermatium to another on the same leaf. That is, remember the plus and minus strains. Okay, that means the Spermatium produced from the same Spermagonium is not able to fuse with the flexuous or receptive hyphae of the same sori, because these are not opposite strains. That's why it needs the insects to spread Spermatium or Basidiospores. Clear? By the time the drop of the nectar exudes, the receptive hyphae grow out through the ostiole into the nectar, and these are spermatized by Spermatia of the opposite strain brought there by visiting insects. Clear? So now it is plus. Now the Spermagonia or Basidiospores which are from the minor strain is reached the flexuous hyphae or receptive hyphae as a result of the insect visitors. So the intervening walls of the point of contact between the Spermagonium of one strain and the receptive hyphae of the another strain dissolve, and as a result spermatization occurs. So the Spermatium nucleus passes through the opening into the receptive hyphae, in which it moves downwards through the pores in the septa. There is no karyokinesis, only that nucleus passes through the Flexocyphae, because the intersecting wall or septa of the Flexocyphae is with the centropause. So it freely moves towards the down area of the this sporangia. See this figure, it is with here one spermatium, here it is attached with, that is of opposite strain. These are the spermatia of the same sorus. So the attached spermatium with the receptive hyphae, at the point of contact normally the walls dissolves and the nucleus from the spermatium passes through the receptive hyphae and reaches the lower portion of the this Sparmagonium. Okay, that is the process. Then what happens? Spermatization, now it is over. This is the actual photograph. See the structure of this Sparmagonium. So finally the spermatial nucleus reaches the basal cell of the receptive hyphae, which thus comes to possess two nuclei which lie side by side in a pair. How it is two nuclei is there? Because the receptive hyphae is also with a nucleus. So they are again dikaryoticed. They won't see any fusion immediately, only spermatization, that is the mixing plasmogamy, that is mixing up of the cytoplasm only. And this pair of nuclei of the opposite strain is called a dikaryon. Now it is called dikaryon. That is, one nuclei is of the same Sparmagonium and the other one is from the other strain of the Sparmagonium. So here now the basal cell, now it is with a dikaryotized and with a nucleus of opposite strain, A plus and minus. The cell possessing the dikaryon is said to be diplodized. Okay. Then as a result of the spermatization, the basal cell of one or more receptive hyphae in the Sparmagonium become dikaryotized. Now what happens? This photograph is also that close-up view of Sparmagonia or Pycnia, and receptive hyphae is there, and so many spores are there. See this, it is attached with the spores. So from this account, it is evident that the Spermatia and the receptive hyphae have taken over the sole function of sexual reproduction, because this Spermatia or Basidiiospores is considered as male cells, only considered as male cells, because the fungus is very lower group of organisms. That so we can consider Spermatia as male cells. And here plasmogamy or spermatization is actually happened, and diplodization, we can consider it as diplodization. Then dikaryotization may also occur when haploid hyphae of two mycelia of opposite strain come in contact. Then the next stage, that is ACL or acidial or ACL stage. That is the haplomycelium, which has built up Sparmagonia beneath the upper epidermis, has the meanwhile penetrated the entire leaf and reached the lower surface. Now from the upper hyphal formation, it will normally reach to the lower surface of the lower epidermis of the leaf. There the mycelial hyphae accumulate and form spherical masses and masses of uninucleated cells. These are called the acidial or ACL primodia. The acidial primodia or proto-acidia lie singly in the substomatal chambers or in the intercellular spaces. Okay. Then some interesting factors are there. See, this is that particular stage. Upper, that dotted line represents the nectar, and here we can see that hyphae and reaches the lower surface, and see the interesting factors about this. So the basal cell of the acidial cup, the lower fungal sporangial area is called as acidial cups or acidial sporangium. On lower surface, the same enlarged surface, the basal cells of the acidial primodia becomes binucleate. See, In the Sparmagonium, there also it formed the binucleated condition due to the spermagonial nuclei passes through the flexual hyphae and reaches the lower cell. But there is no evidence that that same binucleated cells of the this Sparmagonium is not producing any hyphae and reaches the lower nucleus. Then how this takes place, that is the how the primodia which is developed acidial primodia or acidial first formed hyphae produce, how it becomes binucleated, nobody knows. How this takes place is not clear. Okay. So in the mycelial hyphae, they travel passing down through the septal perforations. Finally, this spermatial nuclei reaches the cells of the basal layer of the acial primodia, which thus becomes binucleated. That may be the reason. Okay. And these binucleated cells of the basal layer are called the Basidiospore mother cells. See this, anyway it becomes binucleated and which reaches the lower portion of this acidial sporangial area or acidial cups, and the binucleated cell act like what? This binucleated cells of the basal layer are called Basidiospore mother cells. They divide producing alternatively Basidiospore and intercalary cells. It continuously like that of the spermatial formation. Here also this stalk, that is the basal cell, it is binucleated and through mitotic division produces the polygonal Basidiospore, then form an intercalary cell, and it is normally sterile cells, and again one more Basidiospore. Alternatively, the base Basidiospore mother cell produces Basidiospore or Basidiospores alternatively with the intercalary sterile cells. Okay, intercalary sterile cells. This is the actual photograph of this acidial cup. Here in the lower portion like that of the Spermatio, here also this basal cells which are binucleated, and it produces, here intercalary cells are not visible. Normally it dries out, and as a result the Basidiospores may release. At the time of Basidiospore formation, the binucleated Basidiospore mother cell increases in length. The two nuclei in it divide conjugately. A small daughter cell is then cut off, and it is terminal end. At its terminal end, two of the nuclei remain in the Basidiospore mother cell, and the other two passes into the daughter cell. Likewise, through mitotic divisions, it cut off daughter cells, increases in size and divides into two, upper bigger binucleate Basidiospore and or Basidiospore cell, and lower a small binucleate sterile disjunctor cell. It is otherwise called disjunctor. As a result of cell division, produces a fertile Basidiospore and a disjunctor or the intercalary cell, which is sterile. Okay, likewise, Basidiospores are developed. See this is the photograph, micro slide photograph. That is upper portion we can see this spermagonial formation, and lower we can see lot of acidial cup formations. See, that is brightly colored area. So each Basidiospore mother cell undergo a series of such divisions, and through this way closely packed chains of cells are formed in the tip of the Basidiospore mother cells. The oldest cell at the top of the chain and the youngest cell adjacent to the basal cell or mother cell. Okay. So in meantime, what happens? The marginal cells at the base of the young Basidium divides repeatedly to form a wall and completely surrounds the cells of the Basidium. This wall is called a Paridium. Here we can see this U-shaped lightly colored Paridium or wall formation, peridial formation, and as the Basidia matures, the chains of the cell grow up as a result of this pressure exerts, and what happens, this Paridium breaks and form this cup like structures. Okay. and the ascidial cup partially within the leaf tissue and partly projects outside. So these are all already I told you, and these are for your notes. So the Basidiospores in the chain become separated from each other and fills up the ascidial cup. Then Basidiospores is now exposed, and it is what is a shape polyhedral, thin walled unicellular but it is binucleated structures. So these Basidiospores or Basidiospore actually infects what? The first wheat. That's why our class starts from the Basidiospores. Clear? Then these are the yellow bumps. That is, yellow bumps are due to that upper surface, that is Spermatia, Sparmagonium, that is yellow bumps are there. Now here this one is, now it is exposed, and so we can see the nectar, glossy one drop of nectar is there, and germination of pseudospores. The binucleated pseudospores are carried by air currents and to the primary host, on which they germinate under suitable conditions. At the time of germination, the pseudospores put out a germ tube, a presorium, and likewise the story repeats. Okay, and this is the life cycle, and this is the first part, that is this haploid, haploid, it is not haploid actually, dikaryotic, so it is N plus N. The two nuclei of plus strain and minus strain, Pseudospore infects wheat. There it forms the uridial stage. Again we can see a mini cycle here, that iridospores again infects this. See, the surface of the iridospores are pricky, and it again infects the wheat and repeats or spreads the disease. And now it is that red rust stage is now turned into black rust stage, that is Telia or Teletospore formation. Here we can see the spindle shaped teleospores, and there again infects, and that is now this teleospores in the Teletosauri, and from there the Teletospores germinates to form Basidiospores. Here we can see the Basidiospore. Basidiospore becomes the haploid structure. Here we can see only fusion, but that nuclei is also with that plus and minus strain is there, because it is infected by Basidiospores. Then Basidiospore formation. Normally Basidiospore formation infects the Barbary leaf. Here we can see that yellow bumps on the upper surface, that means pycnidal formation, and from there we can see the pycniospores. This is the lower surface. Lower surface we can see this what SCO, this Basidiospore formation or ascidial cups, and normally this ascidial cups protrudes, that is this figure, that is here we can see the protrusions, and it is the actually this peridial walls we are now it is visible here like that of small protrusions, and inside we can see this Basidiospore formation. Again it infects the wheat. This is a life cycle, and it is macrocyclic because due to the presence of an elaborate life cycle. And here it is, that is once again for your exam point of view, you can see this from here, that is the grass host or the here wheat plant, Pseudospores, that is black and white, that is plus and minus nuclei is there, and it infects. And we can see the sub life cycle, that is iridospore formation, and again it infects the wheat plants for the spreading of the disease, and slowly the same sore becomes Teletosauri. Okay, bicelled condition, then that germ pore formation, and then Basidiospores of plus, it infects the Barbary leaf. There we can see first pycniospores or Spermatium. Then this is a Flexocyphae, Pycniospore or Spermatia. There we can see this Basidia, again Pseudospores, that life cycle repeats. So this is the it's a somewhat large class, nearly 1 hour. So try to study, and here it is for some points. that is the life cycle of Puccinia graminis, and stage 0, that is Sparmagonia bearing Spermatia and receptive hyphae, stage 1, that is Essia bearing Basidiospores, then stage 2, Uridia bearing Iridospores, because Basidiospores actually infecting the wheat. Stage 3, the Telia, stage 4, then again forming this iridostages, stage primary host on wheat, then some a little bit points. So here we can expect so many questions, and starting from the first introductory part, that what is heterogeneous fungi, what is polymorphic fungi, then Spermatium, structure of Teletosauri, structure of Iridospore, then what is this acidial cups. Okay, likewise so many questions we can get from this particular part, and try to study repeatedly, viewing these slides, and try to study by writing the or remembering all these stages, and try to view this life cycle. Okay, and if you are with any doubts, you can ask. And what is the meaning of Presorium, and likewise many questions are there. Okay, try to study, and thank you for patient listening. Thank you.